We have seen that the meninges form the wrappings that cover, protect, and support the brain and the spinal cord, and also provide anchorage for blood vessels that are in close contact with the central nervous system structures. We also see two other protective mechanisms that we'll investigate here. So we have the cerebrospinal fluid and the blood-brain barrier. Cerebrospinal fluid serves as a mechanism of physical protection for the central nervous system, while the blood-brain barrier provides chemical protection for the brain and the spinal cord. So we'll focus on cerebrospinal fluid first and then return to the blood-brain barrier. So cerebrospinal fluid is very similar in composition to blood plasma. So that is, it is mostly water and contains a little bit less protein, a little bit less calcium and potassium than blood plasma, but it does have more sodium chloride than blood plasma. And the reason that it's so similar in composition to blood plasma is because cerebrospinal fluid is produced by small capillary clusters that are located within the brain, and we'll see those in just a second. The cerebrospinal fluid, as mentioned previously, functions in physical protection, so it functions as a cushion to support the brain and the spinal cord. And for the brain specifically, it really helps prevent the brain from crushing itself under its own weight, because the glial cells and the neurons of the brain are very delicate, and the brain is a very large structure comparatively, and those neurons and glial cells are not really built to supporting the weight of the brain. And so cerebrospinal fluid functions as a fluid cushion to help prevent the brain from becoming damaged under its own weight and mass. So the majority of the nourishment for the central nervous system comes from its rich blood supply. However, cerebrospinal fluid does function in a small capacity to provide nourishment to both the brain and the spinal cord. As I've mentioned previously, the cerebrospinal fluid is formed by these structures called choroid plexuses, which are small clusters of capillaries that are found within these chambers in the brain called the ventricles. The choroid plexuses produce cerebrospinal fluid by the mechanism of filtration, so that as blood pressure forces or pushes fluid out of the capillaries and some of the substances that are within that fluid, but it does not push any of the leukocytes or any of the erythrocytes out of the blood plasma. So by filtration, we saw that the fluid is pushed out of the blood vessels, but the cells will remain within the blood vessels. So in a healthy brain and spinal cord, we will not be getting blood cells pushed out into the cerebrospinal fluid. So we also see that cerebrospinal fluid, at any given time, the total volume of it within the ventricles, that is the chambers within the brain, and even the subarachnoid space, which is surrounding the brain, we see that the total volume of cerebrospinal fluid in the central nervous system at any given time is about 150 milliliters. And we also see that about 500 milliliters of it is produced daily. So this means that the entire volume of cerebrospinal fluid is replaced roughly 2.5 times per day. As I mentioned, the cerebrospinal fluid is produced by choroid plexuses, which are small capillary clusters within these chambers in the brain called the ventricles. And we're going to take a look at those ventricles now, but you can see in this coronal or frontal section of the brain, we can see the chambers quite easily because they're highlighted in blue here. And we'll see that these chambers are interconnected with one another, so that cerebrospinal fluid that's produced within them can flow easily from one chamber to the next. So we'll start with the ventricles that are within the cerebrum, and these are referred to as the lateral ventricles. So these are C-shaped ventricles. There's one lateral ventricle in each of the cerebral hemispheres. So we have a right lateral ventricle and a left lateral ventricle. And these two ventricles are connected to what is called the third ventricle that is located between the two halves of the diencephalon. So within the cerebrum, we have the lateral ventricles. in between the two halves of the diencephalon, we have the third ventricle, and these lateral ventricles are connected to the third ventricle by a small canal that's called the interventricular foramen, so that we see that there is one interventricular foramen that connects each lateral ventricle to the third ventricle. We also see that in the kind of making our way inferiorly, we see that there is a diamond-shaped fourth ventricle that's located within the brainstem. This fourth ventricle is connected to the third ventricle by way of the cerebral aqueduct, so that way cerebrospinal fluid in the third ventricle can flow easily to the fourth ventricle. In the fourth ventricle, we see that it has small pores in it, and these pores are called the apertures. We have two lateral apertures on the lateral sides of the fourth ventricle and a median aperture that's located posteriorly on the fourth ventricle. And these apertures allow cerebrospinal fluid to exit the fourth ventricle and enter the subarachnoid space. We also see that at the inferior side of the fourth ventricle becomes what is called the central canal. And the central canal is a small canal that runs in the middle of the spinal cord. So cerebrospinal fluid can continue down into the center of the spinal cord, and it can also leave the fourth ventricle and occupy the subarachnoid space surrounding the brain. The ventricles are lined with a special type of glial cell. They're called ependymal cells, and ependymal cells have cilia on their surface, and this helps to circulate the cerebrospinal fluid within the ventricles. Now, in order to leave the subarachnoid space, so remember the cerebrospinal fluid is going to exit the fourth ventricle through its apertures and occupy the subarachnoid space. And from the subarachnoid space, the cerebrospinal fluid can drain into the dural venous sinuses by way of these small extensions that the arachnoid motor has that are called arachnoid granulations. Now, these arachnoid granulations project from the subarachnoid space into the dural venous sinuses. So in this diagram here, we see that these arachnoid granulations are projecting through the meningeal layer of the dura mater into the dural venous sinus that's called the superior sagittal sinus. So it is through these granulations that cerebrospinal fluid can drain from the subarachnoid space into the blood that occupies the dural venous sinuses. This dural venous sinus happens to be the superior sagittal sinus, but there are other dural venous sinuses and other arachnoid granulations that we can't see in this diagram here. So these are the only ones that are shown, but there are others. There are other dural venous sinuses that sit between the two layers of the dura mater. So we've seen the cerebrospinal fluid, the chambers that it occupies, and the structures that create it. So now we'll turn our attention to the final protective mechanism of the brain, and that is the blood-brain barrier. So the brain itself, and really kind of the central nervous system as a whole, depends on a very constant internal environment. And so the blood-brain barrier helps to prevent a large shift in the chemistry of the central nervous system, because what we end up seeing is that blood chemistry, although it is maintained in homeostasis, its chemistry does tend to fluctuate with things like meals, being overly hydrated, being dehydrated, and things like that. And so the blood, even though it is maintained in homeostatic levels, it does shift quite a bit in terms of its chemistry. And really what the blood-brain barrier is doing is helping to prevent the brain and the spinal cord from seeing those large shifts in chemistry. The blood-brain barrier itself is created by tight junctions that exist between the cells that line the capillaries of the brain, and to the same extent to the ones that line the spinal cord as well. So we see that capillary cells within the brain in particular are also regulated by astrocytes, which are in close proximity to the capillaries, and another type of cell that are called pericytes. And so these two cell types really help to regulate the permeability of the cells of the capillary. So that is, they regulate how porous or how easy it is for material to cross the capillary cells and enter the brain tissue. So because of the presence of the tight junctions, and I'll be focusing on this diagram on the right here, so these are brain capillaries, and most other capillaries are located on the left side of this diagram here. So these are typical capillaries located within a number of places in the body, but these are the kind of the typical configuration of brain capillaries here. So because of the presence of these tight junctions between the capillary wall cells, the presence of the astrocytes, and the presence of the pericytes, we see that the central nervous system capillaries are much less permeable than other capillaries in the body. So this means that anything moving because of the presence of these structures, this means that anything moving from the blood into the brain in these capillaries has to pass through the plasma membranes of the cells in the capillary wall. Whereas in most other capillaries, there are small gaps between the cells that line the capillary walls, and so it's easy for fluid and other substances to essentially squeeze between the two cells and get into the extracellular space that's surrounding the interstitial fluid that's surrounding the capillaries. So returning to the brain here in the brain capillaries, we see that most lipid-soluble substances, things like fatty acids, oxygen, carbon dioxide, alcohol, nicotine, and anesthetic drugs, can very easily pass through the cells that make up the walls of the capillaries and into the brain tissue, because these substances are lipid soluble, which means they can diffuse directly through the plasma membrane. So however, most water soluble substances, many cancer drugs and things like that, are not easily able to pass through the walls of the brain capillaries because they are not lipid soluble. So because we see that a lot of cancer drugs are unable to move through the plasma membranes of certain cells, this helps to explain why treating brain tumors with things like chemotherapy can be particularly difficult, simply because that drug does not have a way to get into the brain tissue. We also see that the membranes of capillary cells in the central nervous system, and again in particular in the brain here, also contain a number of enzymes that destroy chemicals like epinephrine and norepinephrine, which are neurotransmitters, and these help prevent brain neurons from becoming activated randomly by these chemicals, because we see epinephrine and things like epinephrine and norepinephrine being produced in other parts of the body, and they can enter the bloodstream. And so this really helps prevent the brain from becoming randomly activated by these chemicals when the brain was not initiated to do so by neurons or other brain cells. So in certain parts of the brain, I think it's important to note here, like the region of the medulla oblongata that controls vomiting, the blood-brain barrier is almost entirely absent. So that part of or those capillaries that exist near that structure look pretty similar to most other capillaries in the body, where they're quite highly permeable. And so the reason for this is that this allows the medulla to closely monitor the blood for any potentially harmful substances, and this will allow the medulla to trigger vomiting if certain substances are detected. Other locations where the blood-brain barrier is particularly weak are places like the hypothalamus, and we saw that the hypothalamus controls things like water balance, body temperature, and a number of other metabolic activities. And having a lack of a blood-brain barrier there means that the hypothalamus can more closely monitor blood composition.